Donc, encore une fois, bienvenue, Dylan. Merci d'avoir accepté notre invitation. C'est vraiment un grand plaisir pour moi de faire l'introduction de Dylan aujourd'hui. Donc, comme vous le savez, parce que c'est indiqué sur sa présentation, il est professeur au département de biologie à Concordia. Donc, il est prof depuis 2009 à Concordia. Donc, son parcours avant ça, il a fait son bac à l'Université de Guelph, déjà dans le domaine des pêches. Et j'ai eu le bonheur que Dylan choisisse mon laboratoire pour venir faire son doctorat de 2000 à 2005. Donc, ça a été le début d'une belle aventure et ça a été le début pour lui de programme de recherche à long terme en collaboration étroite avec la Nation CRI de UHC. Donc, il a fait son doctorat là et il a continué après son programme de recherche quand il a eu son poste avec eux, donc depuis un bon bout. Et après son doc, il a fait, et d'ailleurs, avec son doc, il s'était mérité le prix c'était le prix Cameron de la Société canadienne de zoologie pour la meilleure thèse, en tout cas jugée puis évaluée par la Société canadienne de zoologie en 2006, que tu avais eu ce prix. Il a fait ensuite de ça un postdoc. Il a été 4 années à Dalhousie. Vous travaillez avec Jeff Hutchings, que la majorité d'entre vous, pour ceux qui sont dans le monde des poissons, connaissez. Mais en fait, il a eu aussi un statut avant Concordia, il a eu un statut de professeur adjoint à l'Université d'Dalhousie à Halifax. Voilà, donc Dylan mène en fait, il est vraiment associé, je dirais, son étiquette première, c'est de façon très globale, c'est la biologie de la conservation. Je dirais qu'il est clairement une vedette dans le domaine de la biologie de conservation au Canada, puis bien au-delà. Puis je dirais même une vedette encore montante. Sky is the limit pour Dylan. Il a des horizons de recherche très larges dans le domaine de la conservation de la biodiversité. Donc, son objectif général de recherche, on peut le définir comme essayer de comprendre ou d'étudier et comprendre comment la diversité des populations au sein des espèces apparaît, évolue et persiste face aux changements environnementaux naturels, changements environnementaux induits par les activités humaines et les conséquences de ces changements-là et de ce maintien ou pas de la diversité génétique sur la conservation et la restauration des espèces, avec une emphase sur les espèces aquatiques en lien avec la gestion des pêches, mais quand même, je dirais, avec une vision plus globale sur la conservation de la biodiversité à l'échelle globale et même mondiale. S'il était à l'IBIS, on dirait qu'il est un fit parfait pour être à l'Institut de biologie intégrative, parce que clairement, son approche de recherche est une approche très intégrative avec du travail strictement, en fait, de l'écologie pure, lié à la génomique, lié à du travail expérimental en laboratoire et dans une perspective sur des bases conceptuelles théoriques solides, mais très, très souvent et même en général avec une vision sur les applications. Puis, je dirais aussi, en fait, d'une certaine façon, Dylan nous force à réfléchir sur nos bases acquises sur les grands principes de biologie de la conservation, notamment sur jusqu'à quel point les populations de petite taille sont vraiment mal foutues ou pas d'un point de vue chance de survie ou pas. Donc, il a fait des papiers, je dirais, des papiers qui ont été marquants à ce niveau-là, puis qui nous forcent à réfléchir et voir les choses un peu directement. Puis j'imagine qu'il va nous en parler un peu aujourd'hui. C'est un chercheur prolifique. Il y a déjà une centaine de publications. Mais ce qui est marquant, mis à part ses contributions empiriques très, très solides, Dylan, il est passé maître dans l'art de faire des papiers de revues très conséquents et des papiers de méta-analyse aussi. Et c'est des papiers qui sont très influents, abondamment cités. Donc, son tout premier, en fait, qui avait fait son travail de recherche documentaire quand il était au doctorat ici, puis qui a été cité plus de 700 fois, qu'on utilise dans nos cours maintenant sur une vision plus adaptive ou adaptative du concept de définition d'unité de conservation. Adaptative dans le sens de rester flexible sur les critères qu'on utilise pour définir des unités de conservation pour s'adapter au contexte précis dans lequel on veut mettre en application ces choses-là. Donc, c'était déjà une vision qui se démarquait de cette espèce de guéguerre qui existait à l'époque, qui existe encore jusqu'à un certain point, de dire c'est moi qui ai la bonne définition de qu'est-ce que c'est qu'une unité significativement évolutive, etc. Dès là, sa perspective, c'était wait a minute, ça dépend du contexte, il y a du bon dans tout et le but, c'est de réussir à conserver, c'est pas d'avoir une approche dogmatique. Et il a fait un papier aussi de revue super important sur l'ampleur et l'échelle de l'adaptation locale, focusé sur les poissons, mais avec un intérêt beaucoup plus large que ça pour les gens même qui ne travaillent pas sur les poissons. Et des travaux sur les programmes de reproduction en captivité et j'en passe. Donc, encore un jeune chercheur, très, très influent dans le domaine. Puis, on a bien hâte d'entendre ce que tu vas nous raconter aujourd'hui. Merci, Dylan.
Merci beaucoup pour votre introduction, Louis. Ça m'a fait plaisir de partager notre recherche avec vous aujourd'hui. Je ferai la présentation en anglais, mais n'hésitez pas à me poser des questions à la fin de la présentation en français et on peut avoir une bonne discussion en anglais ou français ou l'autre. Hi, everyone, and thanks for the invitation to share my team's research with you all today. I really hope everyone is safe and healthy. And before I get started, I just want to acknowledge the very significant contributions to the research I will share today of my graduate students and postdocs. Much of this is their thesis work, and it's been a pleasure working with them on questions relating to small population biology over the last several years. One of my lab's major research avenues is understanding how species adapt to environmental change. We all recognize that we live in an era of unprecedented human-induced change. Habitat loss and fragmentation, of course, poses one of the greatest threats to the survival and the persistence of many species on our planet. It leads to small population size, and it traps these populations in small, isolated fragments. Small population size increases the likelihood that random events, such as extreme weather, will wipe a population out altogether. But small population size also generates genetic issues that may affect population persistence. Now, in general, to adapt to environmental change, species can do three different things. First, species can simply disperse to a new, more hospitable habitat. But as I just noted, this is impossible in many instances due to human-induced habitat fragmentation and isolation. So as a result, many species have two other options. One, they can either evolve through adaptive evolution, where certain genetic variants are favored and allow persistence, but this requires genetic diversity, which, as I will point out on the next slide, is expected to be in reduced quantities if the population size is small. Or two, species can respond to change through phenotypic plasticity, the differential expression of the traits it possesses in relation to environmental settings. Now, persistence via phenotypic plasticity is also expected to benefit from at least having some genetic diversity underlying traits. Now, small isolated populations are expected to have reduced genetic diversity, and thus small populations are expected to have reduced adaptive potential, because genetic diversity is considered the raw material for allowing a population to respond to environmental change. Small populations may have reduced phenotypic plasticity as well, given that the processes that reduce genetic diversity underpinning trait plasticity operate more in small populations. So when populations are isolated and cannot exchange gene flow to replace genetic diversity quickly, they lose genetic diversity through random genetic drift that occurs disproportionately at a small effective population size. Now, NE, this effective population size, is how many adults contribute offspring to a new generation of the population. NE is usually much smaller than the actual number of individuals in a population. Inbreeding is also an inevitable consequence of small effective population size, and the consequence of increased inbreeding is greater inbreeding depression, which is usually a reduced ability to survive and reproduce. So as a result, it's commonly thought that a population shift to a small fragmented state results in an extinction vortex, wherein a loss of genetic diversity and inbreeding resulting from this low effective population size reduces adaptability and survival, which further reduces the census population size, which further accelerates the genetic consequences of small NE, and so on. Some authors have therefore concluded that large population size is essential for adaptive responses to environmental change. So along with the extinction vortex concept in conservation biology has been development of a simple prescriptive rule about what minimum viable effective population sizes should be to ensure population persistence. General recommendations have been made that an NE of 50 is sufficient to prevent the deleterious effects of inbreeding depression in the short term, so over several generations, whereas an NE of 500 is required to maintain the long-term adaptability of a population in perpetuity. So this so-called 5500 rule. There remains a lot of debate as to whether this rule is generalizable and sufficient, with some authors actually recommending it be shifted to the 100 to 1000 rule, given new data on the prevalence of inbreeding depression in the wild over the past few decades and how the original 5500 rule was formulated. Interestingly, though, the actual relationship between genetic variation and population size in nature still remains largely unresolved. So single locus and polygenic models of the relationship and how it relates to the type of selection lead to quite different predictions about how much effective population size must be reduced before much genetic variation is lost. So typically, population genetic models, which are usually single locus, predict that a population needs a higher effective population size to retain genetic variation. Traditionally, many studies have used single locus DNA marker assays of neutral genetic variation, i.e. variation not under selection, to infer adaptive potential. But we know that these assays rarely, if ever, correlate with genetic variation underlying quantitative traits, and they do not account for how selection acts on genetic variation. Furthermore, empirical studies testing the theoretical expectations of reduced adaptive potential in small populations are remarkably scarce in nature. So much of what we know of how effective population size affects either adaptive or fitness responses is restricted to lab studies of bacteria and fruit flies. Now, of course, these species have practical advantages for experimentation on replicated populations over successive generations. But the environmental conditions of these lab studies do not typify the heterogeneity that most species experience in nature, nor are these species' biological characteristics necessarily common to species of conservation concern. So the extinction vortex can occur, and there are many good examples that now exist showing how small population persistence can be potentially enhanced through interventions such as genetic rescue, in which individuals from healthy populations are introduced into the focal population to replenish genetic diversity and to boost fitness. But once one considers natural selection in this loop, there are good reasons to expect that for some species and under some environmental conditions, selection can either dampen the classic genetic effects of small population size, such that it takes much longer for the vortex to occur, or selection might actually maintain or even enhance adaptability in some small populations, effectively rendering extinction dependent on non-genetic issues, at least on timescales that matter for conservation biology. So in this talk, I will attempt to bridge some current knowledge gaps regarding small population responses to environmental change by overlaying results from a series of observational, common garden, and field experiments conducted on the same system of natural populations. So first, I want to highlight that selective pressures change during the habitat fragmentation process. Second, I want to emphasize that many different environments are associated with small population size in nature. Small populations do not necessarily by default inhabit marginal or poor habitats. Third, I want to illustrate that some small fragmented populations with effective population sizes that would be below minimums commonly adopted in the conservation biology literature can be important sources of genetic diversity that can be capable of plastically responding to environmental change and capable of long-term persistence and perhaps adaptation. Now, these findings provide some hope for some small populations, but there are contact dependencies and contentious issues to consider. And lastly, I'd like to share two other research avenues besides small population biology in which my lab is applying genetic and evolutionary principles to fish and wildlife conservation. Okay, for three reasons, our lab conducts collaborative research on salmonid fishes to address questions relating to adaptation to environmental change and small population conservation. So first, these are socioeconomically and culturally important species for a variety of fisheries. Second, these species exhibit substantial population differentiation and local adaptation. So these attributes combined with the spatial discreteness of their populations makes them really useful models to investigate the role that population variation and genetic variation may have on influencing responses to environmental change. And finally, salmonids are experiencing considerable declines in many parts of their native ranges. So a better understanding of their population responses to environmental change can not only inform conservation efforts for these species, but other vertebrates as well. So our study species for today's talk is the brook trout or brook char, an endemic of eastern North America. This is a species that thrives in cold headwater streams and lakes with few other competitor species. So brook trout have one of the highest levels of population structuring known for vertebrates. Literally tens of thousands, if not more, of this species, populations of this species exist within Canada. And in the southern part of the range of its native range, many brook trout populations have become fragmented and extirpated by human activities. But it's important to note that in many parts of its northern range, brook trout populations have always been naturally fragmented in streams and lakes that became isolated after the last deglaciation. So we studied the genetics and the evolution of naturally fragmented brook trout populations situated in Cape Race, Newfoundland. A main benefit of studying populations here is that the trout live in very small streams, which terminate as 30-meter waterfalls directly into the Atlantic Ocean. And that means that population isolation can be confidently confirmed. The trout populations are also free of any anthropogenic influences like fishing or fish stocking. And the reason for this is because the trout are tiny. Nobody really cares about them. So adults commonly in these streams are only 4 to 6 inches long. Furthermore, the small size of these streams allows us to comprehensively sample populations to confidently determine that they vary substantially in terms of their abundance from only a few dozen adult individuals to several thousand. Now, N here in these images relates to the census population size, whereas NB is the effective number of breeders. And the effective number of breeders is basically the effective population size, but for a single age cohort, i.e. how many adults produce offspring to each new annual cohort within the population. So a main point is that several of the populations we research here have an effective population size that would translate into values that would be lower than 50 in nature. Okay, so as in other parts of the species range for brook trout, there's pronounced population structure within the system, with stream drainages big and small, all being distinct from one another and easily demarcated using tens of thousands of single nucleotide polymorphisms. The best available evidence that supports that populations originate from a common ancestor and that most populations involved in our research have been isolated for at least a few to several thousand years. The demographic histories of Cape Race populations using these stairway plots also suggest that populations in different drainages experience these dramatic declines in effective population size approximately 8,000 years ago. So this time frame corresponds to when the main fragmentation of contemporary stream drainages took place following landscape rearrangement that was associated with deglaciation, with much smaller, subsequent smaller population size declines towards what contemporary sizes are approximately 2,500 years ago. So just for your general information, the average generation time of brook trout on Cape Race is about 2.2 to 2.5 years. And so most of these populations have been isolated for at least 1,000 generations or more. Okay, so a number of populations, particularly within the central region of the Cape, with the most extreme habitat fragmentation and the smallest population sizes, have accumulated high frequencies of deleterious mutations at several genes, just highlighted here in yellow. Now, these genes are associated with oxidative stress and other cellular functions. And although we have not confirm that these mutations affect fitness within brook trout populations per se, the collective results here provide a hint that small populations may carry some maladaptive characteristics, yet persist for long periods of time. So interestingly, small populations show evidence of having local adaptations to their respective environments, despite having disproportionately less neutral genetic variation, i.e. again, that variation that is not under selection, when we compare them to large populations. So these figures show the percent of coding region single and nucleotide polymorphisms linked to functional traits in brook trout that are putatively under balancing or divergent selection, as outliers differentiating population pairs of various population sizes. Signatures of selection were present at adaptive loci in all small populations surveyed. The greatest signatures for selection were observed amongst small versus large populations or among small population pairs. There are also many phenotype environment associations among Cape race populations in terms of body morphology, coloration, spot pattern traits in both sexes, consistent at least with the hypothesis that selection may be influencing interpopulation differences in phenotypic trait differentiation. At a number but not all traits, we also see this evidence for a greater spread of trait values at the small population size end of the spectrum along Cape race. So the overall greater variability among small populations is consistent with what is observed or what is known of habitat variability in Cape race streams. So small populations do in fact inhabit small streams, as shown in the bottom middle panel here, but small populations have more variability in habitat means and in coefficient of variations for these habitat means for many different habitat parameters that influence growth and survival in brook trout. The ones highlighted here are pH, mean stream temperature, the mean stream depth, and the mean stream velocity. Now, more recently, we've revisited adaptive differentiation amongst Cape race brook trout population using a richer set of single nucleotide polymorphisms. We've specifically used redundancy analyses to better understand how gene loci under selection may relate to different abiotic and biotic environmental variables within these different streams. This is still a work in progress, but the results reiterate the point that small populations exhibit signatures of adaptive differentiation and that several abiotic and biotic factors are implicated. Of particular note, although in a number of streams, brook trout is the only fish species present, in some they share stream habitats with landlocked Atlantic salmon, which is another salmonid fish. And the red circle here is just to denote that the presence of salmon appears to be among the variables associated with adaptive differentiation in the trout. So despite being situated at a very fine scale, you can see tremendous trait differentiation among populations. So you can go cross over, do a hike from one stream to the next. They can only be 300 meters apart in the wild, and you can observe up to a threefold difference in mean trait differences at the same age, whether you observe them in the wild or whether the fish are reared under common environmental conditions in captivity at our aquatic wet lab facility here in the heart of Montreal. The differences observed when populations are reared under common environmental conditions supports that the population trait differentiation that we observe as a genetic basis. Now, as I mentioned, as expected, small brook trout populations do have disproportionately less neutral genetic variation than large populations using single nucleotide polymorphisms across the genome. However, when we generate large numbers of full and half sibling families within populations, and we then rear them under common environmental conditions in a wet lab, we found no evidence for a positive relationship between additive genetic variance at different traits and population size. So whether life history, morphological, or behavioral traits were assayed, small and large populations had very similar levels of quantitative genetic variation. Now, additive genetic variance is essentially how much genetic resemblance there is between parent and offspring. So higher levels of it allow populations to respond more rapidly to a changing environment. Now, we've also investigated the relationship between population size and the extent of phenotypic plasticity in Cape race brook trout populations by generating large numbers of families from the wild and rearing them in the same environmental gradients. So these figures show how juveniles from different populations and families grow or survive when they were exposed to simulated climate warming. So at the left, population differences in phenotypic reaction norms exist, but they are in general quite subtle and they're not related to population size. And at the right, each dot is a separate family, and basically the average magnitude of plasticity that its members expressed across different thermal regimes. There were no differences among populations in their extent of plasticity. Again, despite inclusion of small populations with an effective number of breeders that would translate into an effective population size of 50 or less. So results from several studies, very importantly overlaid on the same population system, support that compared to large populations, small populations do in fact have less neutral genetic variation, but they retain similar levels of quantitative genetic variance and phenotypic plasticity. They show similar indications of adaptive differentiation at functional loci, and small populations have greater interpopulation trait differences in many instances. That small populations are exposed to greater environmental variability suggests that many different habitats are associated with population size and small population size in nature, and that different small populations may harbor unique adaptive variation. Now, while the results of all the studies I've just shown you are useful, what we really want to know is how small populations, how they respond to environmental change in nature itself. So interestingly, Cape Race is also full of these small, isolated fishless ponds that vary tremendously in their environmental conditions. So we've actually been able to transplant controlled densities of juveniles from different populations to then investigate their responses to a gradient of abrupt environmental change. Now, this basically involves standardized collections and transplants of trout from single populations to independent ponds in the early summer. And then in the early fall, we return back to the ponds, we capture all the remaining survivals within them to be able to quantify trout population survival and growth. And over 4 consecutive years, we were able to transplant individuals from 12 different populations for a total of 97 translocation events. The number of translocations was influenced by the availability of different ponds and by the number of juveniles available in each wild population that would ensure the experiment was not generating a demographic risk to the wild populations. And so with background information on the different populations, we determined the best predictors of population responses to environmental change when they were transplanted, namely the environmental variables of the ponds themselves, the effective number of breeders from each population, the extent of genetic diversity or genome-wide variation within each population, the degree to which each population experienced environmental variability within its past, and the degree of environmental change that each population experienced when it was transplanted, based on the environmental differences between the home stream environment and the new pond environment. Now, interestingly, we found that genetic variables or past environmental history had no influence on survival in new environments. The main influences on survival were habitat variables, chiefly pH and temperature, and maternal effects by initial body size as a young juvenile. So individuals from all populations survived better in less acidic ponds and when they were an initially larger body size. Survival also declined as temperature increased, as would be expected for a cold water species such as brook trout. We thought that an increasingly stressful environment through heightened acidity would disproportionately impact those small populations, and yet individuals from all populations performed equally poorly under increased environmental stress. Growth, individual growth, was also entirely explained by habitat variables, not genetic variables. As expected for a cold water trout species, growth was optimal at temperatures between 12 to 16 Celsius, and growth was higher in deeper, presumably less stressful ponds. Growth here is a reasonable proxy of fitness at this particular life stage because large body size obtained before their first winter increases overwintering survival within brook trout and related salmonid species. So in our field experiment, genetic variables were not linked to fitness under environmental change. It appears that small isolated brook trout populations are capable of long-term persistence and can still respond quite adeptly to abrupt environmental changes in nature. So it raises the question as to why, in general, small isolated populations of this particular species can persist over long periods of time in nature. We suspect that this species is in some senses adapted to living in small populations by being able to overcome some of the maladaptive effects of small population size. The mechanisms for this ability may include evolving in small first-order streams where there are few other competitor species, having a partially duplicated genome, being a highly fecund animal, and past purging of mutational load. Brook trout may not necessarily be an anomaly in this regard. It's important to consider that the history of many freshwater environments, including in most of northern Canada, has been one of serial episodes of fragmentation, isolation, and recolonization through glaciation, deglaciation, and isostatic rebound. Perhaps a number of other freshwater fish species have sufficiently overcome the maladaptive effects of small population size to be able to persist in these kinds of environments. Now, very interestingly, in the past few to several years, we see there are a number of other genomic studies that have also been published on other species, chiefly mammals and other fish, which show evidence for adaptation to environmental change within very small populations harboring low levels of genetic variation, including to novel selective pressures elicited by human activities. Of course, I do want to mention a caveat. 8,000 years of isolation and fragmentation is a long time. And perhaps the argument could be made that many small trout populations at Cape Race became extirpated from those classic genetic effects, and that the remaining ones we are studying now are the robust ones that made it through. I would argue that this seems unlikely given the small geographic scale, but of course we cannot go back in time to refute this possibility. If this was true, though, we might expect to find many small unoccupied habitats without brook trout in the present day, but this is not the case. So we've mapped the detailed distribution of brook trout across all of Cape Race. This is an area that would encompass about a 25 by 25 kilometer square grid. And as alluded to previously, brook trout presence is fundamentally influenced by pond or stream pH. So virtually everywhere you expect brook trout to exist in ponds and streams on the pH that they like, you find them there, including in small isolated habitats. And in the few cases you don't find them, simple explanations exist, such as a pond being too shallow to sustain fish or having a high elevation that rendered a dispersal limitation to get there in the first place. The other intriguing point is that although small and large Cape Race populations respond equally to environmental change in nature, this is not the case when we rear them in novel captive environments in our university's wet lab for a single generation. So here some of the effects of reduced genetic variation are first evident. All populations suffer significant mortality and they have quite poor lifetime reproductive success in the captive environment. But populations with reduced heterozygosity tend to have the poorest performance and experience the greatest phenotypic changes in captivity relative to their wild counterparts. So a main conservation implication here is that the nature of a novel environmental change that a wild population is going to experience will very much likely influence the degree to which classic genetic effects of small population size are a concern. Okay, so overall, our work suggests that small, low diversity populations of some species can be important sources of variation and may be capable of maintaining long-term fitness in and ultimately adapting to changing natural environments. If we can determine which taxonomic groups, species, and even divergent populations that this applies to, certainly the field of conservation biology may be able to significantly improve how it prioritizes its efforts and limited resources. Hopefully our work will stimulate more discussion on whether minimum viable effective population size targets require nuancing. And there's certainly much still to learn on which metrics may predict adaptive responses to environmental change in nature. So our research on brook trout suggests that the utility of commonly used measures, such as neutral genetic variation and additive genetic variance, is likely to be very context dependent. The last point I want to mention is a contentious one for conservation, and that is until populations become very, very small, habitat fragmentation can be a creative process in generating new diversity because selective pressures change as fragments and population sizes are reduced. Now, our research suggests that populations of varying sizes inhabit a variety of environmental conditions. This may make it more difficult to prioritize between populations or to apply general prescriptions and to determine what to do when idiosyncratic selective pressures induce some degree of adaptive change within small populations of endangered species. Now, despite some of the research results on brook trout I've just shown you, I do remain of the view that we need to be concerned about recognizing and conserving genetic diversity in many instances for biodiversity conservation, and that certain taxa may be particularly sensitive to the effects of reduced genetic diversity. My lab is therefore applying genetic principle and evolutionary principles to a number of other research contexts, and one that we're really excited about is the application of genetic diversity to broad-scale conservation planning. So we've recently published a database on nuclear genetic data for vertebrate populations inhabiting the American continents based on 25 years of research that use microsatellite loci as molecular markers. This database basically extends what has been assessed in thousands of studies on genetic diversity within single species conducted at local scales to larger scales through georeferencing and cross-species data synthesis. And as an initial work, the database revealed continental differences in the amount of genetic diversity found within different vertebrate groups without testing possible causes. So observed heterozygosity and mean number of alleles here are both metrics of genetic diversity. And notably, South American and Caribbean vertebrates exhibited reduced values of these metrics compared to vertebrates inhabiting North America or Central America, though the average differences were not that large overall. Now, one way in which we're particularly using the database is to investigate the latitudinal distribution of genetic diversity. So the latitudinal distribution of species richness is, of course, one of the most widely recognized and predictable patterns of biodiversity. Low latitudes in tropical regions have many more species than high latitudes of temperate regions. And that's because tropical regions, or at least thought because tropical regions are more hospitable to a larger number of organisms in being warmer, more humid, more stable over time, and hence more predictable over time. But to date, the extensive attention that's been directed to these species richness gradients has not been extended to understand the broad-scale distribution of diversity within species. And what we're finding overall is we're identifying an uncoupling between species richness and population genetic diversity in many instances due to historical and contemporary factors. So for example, these are distribution maps for species richness versus the two metrics of population genetic richness that I just alluded to, that being heterozygosity and mean number of alleles for terrestrial mammals. And the salient point is that areas which harbor many species do not necessarily harbor the most genetic diversity within species. This discordance has important implications for how we refine broad-scale conservation planning that considers different biological levels of biodiversity and not just ones that focus at the species level. So we're also assessing relationships between human influences and vertebrate genetic diversity in North America, where the most robust data on human land use and genetic diversity exist. And this is being investigated by considering several anthropogenic biomes, such as urban, village, and cropland environments, as well as wild biomes with very little human influence and human population density. And this work is still in progress, but it is revealing some important trends. As we might expect, on average, wild populations of different species found in areas with higher human population density, represented by HPD here, do in fact have lower genetic diversity. Similarly, populations that are wild that live further away from urban biomes, i.e. areas with greater overall human imprints on habitats, tend to show higher levels of genetic diversity. Overall, the trends reveal that humans do have an erosive impact, or suggest they have an erosive impact on genetic diversity within vertebrate species. And this is an important point given that such genetic diversity, of course, is thought to be the raw material that's critical at some level for adaptation to environmental change. You will also note here that there is a lot of spread in relation to increased human population density, hinting that different vertebrates may respond quite differently to higher numbers of humans. So we've investigated taxonomic variation more in depth in mammalian species of North America in relation to both human population density as well as road and traffic effects, because terrestrial mammals in particular are sensitive to road networks and encroachment by human populations. Now, although we find the overall signal is one in which increased roads and human exposure is associated with lower genetic diversity within mammalian populations, the impacts are very species specific. So you have some species like caribou that do not do well with humans or roads very well at all. You have others like bears that show a mix of responses, whereas other species like coyotes actually show positive responses to roads and human population density. So species-specific responses to habitat fragmentation by ongoing road development and human encroachment likely depend on the specific capability to navigate roads as either barriers or as movement corridors, and to exploit resource-rich urban environments. So the non-uniform genetic response to roads and human density that we observe highlights the need to implement efforts that can mitigate the risk to vehicular collisions, while also importantly facilitating gene flow between populations of particularly vulnerable taxa, such as caribou. So another research avenue where my lab applies genetic and evolutionary concepts to conservation is through collaborative monitoring of harvested fish populations in several regions of northern Canada. And one particular region that Louis mentioned in his introduction to me has been where we've been active with the Indigenous Cree Nation of Mistissini, situated on the shores of Quebec's largest lake, Mistassini Lake. Mistassini is a virtual inland sea. It's approximately 2,300 square kilometers, and it's teeming with these large fish from different species, which the Cree have depended on for their survival for several millennia. Now, the community of Mistissini has experienced unprecedented economic and population growth over the past 15 years, and it's opened itself up more to tourism from the south. This has had an impact, or at least a direct influence on the overall fishing pressure on a number of different species, with an increase in these activities, including on distinct populations of walleye that inhabit rivers near the community in the southern part of the lake. So in response to recent community concerns, the Cree Nation of Mistissini initiated a long-term monitoring program of local walleye populations with our lab. The plot on the left highlights a 20 to 40% reduction in the body size that has occurred rapidly over a 12 to 15-year period in both sexes of walleye in the southern populations where human activity is found, but not in a lightly fished northern population. And a survey of genomic change within populations, both before and after the size reductions took place, is at least consistent with the potential for some of the size reduction to have a partial evolutionary basis. Now, fishing here and elsewhere is size selective. That is to say that humans target the largest fish, and body size has a genetic component within fishes. So when large fish are removed, it's thought that populations evolve towards smaller, slower growing fish, as these are the ones that are the ones that survive and reproduce. Now, overall, this is an adaptive response by the fish population to harvesting, but with obvious negative consequences for humans in terms of the potentially reduced biomass of fish that are available for human consumption. So in responses to these rapid changes in Mistassini's fish populations, we complement our scientific research with syntheses of Cree knowledge to facilitate community-based conservation and management strategies for the Cree's harvested fish. And that includes through regular public awareness and education programs. So overall, to kind of wrap up the talk, I think there's certainly much value in considering genetic and evolutionary thinking, not just in terms of thinking about small population biology, but also biodiversity conservation planning and other areas of fish and wildlife management. And with that, I'd be happy to entertain any questions you may have. And thanks for being at my talk today.
Merci beaucoup, Dylan. C'était aussi broad que je l'espérais. Et ça traduit très bien l'introduction que j'ai faite de toi. C'est ton programme de recherche et tout ce que tu touches. C'est absolument fascinant. Puis, et aussi, je le pense profondément, c'est game changer. Donc, c'est ouvert pour les questions et commentaires pour Dylan.